My name is Caitlin McHugh. I'm from the University of Washington, and my fellows project was a mixed model approach to testing multiple correlated traits in large samples. And this was an application to the TOPMed hematology phenotypes. I take genetic data, usually from humans, and apply statistics to those genetic data. And from those, I can answer questions such as, is there a specific disease associated with a trait? In my fellows project, I took genetic data and looked at, is there a specific gene associated with hematology traits, specifically red blood cell traits, such as the number of red blood cells that you happen to have in a sample of blood, or the width of those red blood cells in your blood sample. And by using statistics, I can associate genes with disease. Without my work, which can link genes to diseases via statistical analysis, then it's difficult to design drugs that target the proper gene or molecular part of the gene that we can then use to help cure or treat diseases. So it's through that statistical analysis and that link of genes with traits that we can then unlock new drugs and drug targets. With a cloud compute environment, such as that with BioData Catalyst, I was able to do my analysis in a manner that was not constricted by the traditional compute environment limitations, such as the amount of compute power I had or the amount of memory I had available to me. So BioData Catalyst is really exciting because it allows researchers from anywhere and everywhere to have access to high power compute in order to achieve their research goals. With BioData Catalyst, I was able to have access to the compute infrastructure in the cloud environment so that I could look at more than one trait at a time. And scientifically, this is important because we're able to look at the biology side of things in a more complete manner, rather than artificially choosing one trait of interest and having to look at that. I can model in my statistical method the correlation between multiple traits and then use that to associate with the genomic data. And this allows us to gain further biological insights that we can then use in downstream analyses. I was able to develop code and release it in a streamlined manner in a way that I would not have been able to do without BioData Catalyst. By developing my code within the BioData Catalyst environment, it was already in a state that was suitable to share with other users. The best aspect of working in BioData Catalyst was the community. Being able to have access to users, the help, the support, really feeling like a member of a broader goal and mission. I would recommend BioData Catalyst, and specifically, I would recommend it to researchers who do not have access to compute power in order to achieve their research goals. And this is because of the cloud compute environment that BioData Catalyst offers, that a user has access to higher compute that they might otherwise. Oftentimes, especially with the advent of big data, large compute is required, storage is required in order to truly do the analysis that is important to your research question. Someone should join BioData Catalyst because they have access to this compute. And additionally, by joining, not only will they get those hard resources, but they will be joined into a community of researchers who are motivated to progress the research community.